Hi, it's Catherine. Welcome to Lecture 6. Today we're going to talk about enzymes. I'm going to give you some real-life examples of enzymes, then we're going to go over some practice questions, and along the way I'm going to be asking you questions. So as you go through the lecture, feel free to pause it and write your answers down or say them out loud or do something beyond just thinking about it, because then you'll solidify the information a little bit better in your mind. So say them out loud, write them down, tell your cat, whatever it is, and then unpause the video and I'll give you the correct answer. So kind of like Blue's Clues. Did you guys watch Blue's Clues when you were growing up? Maybe? Okay, so let's get started. Lecture 6, enzymes. Our task is first to think about some real life examples. I'm going to give you 3 digestive enzymes and then my favorite enzyme of all time. Here's our first digestive enzyme. We're looking at amylase. This is amylase in the bottom right corner here. This is the overall structure of your enzyme. So it is the tertiary structure. These spirals here and here are part of the secondary structure. And if I were to zoom in and look at just the amino acids, I'd be looking at the primary structure. What amylase does for us is it takes starch, this big polysaccharide, and it breaks it down into monomers, like monosaccharides. Our monosaccharide in this example is glucose. You're going to see a trend with our enzymes. They're all going to end in ACE, A-S-E, which is our indicator that it is a catalyst protein and it's going to be doing something for our body. Now, since we're talking about digestion, I wanted to include a picture of where you get your starches from. Your starches are pastas and grains and legumes and bread, and bananas have a ton of starch in them, so do potatoes. So really good stuff for your body in there. And in our body, we make amylase in our saliva, in the pancreas, and in the small intestine. What's really cool about amylase in the saliva is it starts working on your food immediately. The moment you put it in your mouth, it starts breaking starch into sugar. So if you were to take a saltine cracker or something and chew on it for a long time, it would start to taste sweet. It would be breaking the starch into glucose, and you can physiologically feel that reaction because you can taste the sweetness. As you swallow amylase, it totally still works. It goes down to your stomach, and then this pH in your stomach denatures it. So it works from the tip of your tongue to just about where it drops into your stomach acid. And then later you'll produce it in the small intestine. Our second digestive enzyme that we get from these beautiful oily avocados and walnuts and salmon and beans and olive oil is going to be lipids. So the enzyme that breaks down lipids is lipase or lipase. I'm going to say lipase. And what it does is it takes our triglyceride and cuts the glycerol off the end, releasing 3 fatty acids. Here are the 3 fatty acids. So it breaks apart our triglyceride into the monomers of lipids, which are glycerol and fatty acids. Here is what it looks like, really beautiful. The alpha pleated sheets are over here, and the beta helices are over here. And our fun fact for lipase is that there's a special kind in breast milk that helps babies digest the fat in breast milk. So it's pretty neat. It's actually in the breast milk, so the babies don't have to produce it all on their own. You're giving them all this wonderful sugar and fat, and you're also giving them the means to break it down. Our last digestive enzyme is protease. Our main one that we use is pepsin. You may have heard of pepsin before. It's produced in the stomach. It's also in the pancreas and the small intestine, but most of where we use it is the stomach. And pepsin takes polypeptides and breaks them into amino acids. So here's our equation here. We have a polypeptide, and our protease is how they spelled it. That's totally cool. Breaks this polypeptide into amino acids. When people think about proteins in their diet, often they think about animal products, And I want to make sure to highlight that you can absolutely get enough in your diet if you eat plant material only. So all the vegans in the class, you're going to be great. Just eat lots of beans and nuts and legumes and you've got it. Also, the nutritional yeast is great. It's delicious. Try it on popcorn. Our image in the bottom right is not a human protease. This is a protease from the COVID-19 virus. And it's really cool because it was recently discovered. And what's neat about this protein, this catalyst right here, is that it's used when the virus replicates. So now that we know what it looks like, we can try to disarm it. We can try to denature it. And if we break it, then maybe we can stop the virus from replicating. Now it's time for my favorite enzyme, and that is tyrosinase. Tyrosinase is super cool because it's the first enzyme that's used to start a biochemical pathway in making cat fur color. So here's my enzyme, tyrosinase. This is the first step. I gotta change it into ubiquinone. That's how that's said. And then from there, I can produce these different molecules leading to pheomelanin, which is the orange-yellow pigment in cats, or lead it through these two processes. There's two different pathways to eumelanin, which is the brown-black pigment in cats. Here's an example of a cat that has a functional tyrosinase enzyme. That's my kitty, little Callie. I want you to take a moment and think, what would it look like if a cat had a non-functional tyrosinase enzyme? And if you guessed that the cat would have no color because the biochemical pathway would not be able to function, you are correct. And that's how we end up with albino kitties. So this adorable albino kitty could have the same pigmentation as my kitty, except this cat doesn't have a functional tyrosinase enzyme and is unable to make fur color. Check it out. The albino cat has pink eyes. These pink eyes show you that there is no pigment in the iris. Instead, you're looking right into the blood vessels. So my kitty has yellow eyes. She has pigment in her iris. And this is just no pigment, but blood vessels, which is super cool. What happens with an albino cat is that the tyrosinase enzyme, this is the active one, in the mutation for albino cats, it gets truncated, which means that it gets cut off too early and it doesn't have the correct shape. So these two do not look the same. This is a non-functional version. This is led to by a frame shift mutation, which is a mutation in the DNA that codes for this protein. Remember, all of our enzymes are proteins. You can end up with albino kitties in a litter with non-albino kitties, and they just are different gene carriers. So two siblings here. This cat has a functional tyrosinase enzyme, and this cat would probably have a similar fur pattern if they have the same father, and has a non-functional tyrosinase enzyme, and thus no colors. Have you heard of Siamese cats before? They're really adorable and they're known for their voices. They talk a lot. So if you have a friend with a Siamese cat, or if you have one, they probably holler at you all the time. They're just chatty cathies. And Siamese cats have a mutant version of tyrosinase, except they're not albino. So it's not non-functional, it's just mutated. And here's the mutation. Their tyrosinase enzyme only works in colder areas of their body. So their toes, their tails, their ears, those are extremities. The nose and face get less blood, less heat than the rest of the body. These areas are able to make pigment because the tyrosinase enzyme is chilly enough for it to work. In the warm places of your cat, it's too warm and the enzyme is not functional, which is super cool because then it explains why indoor and outdoor Siamese kitties look a little bit different. So which one of those do you think is the indoor kitty? If you said the top one here, yes, this is the indoor kitty. And I know it's the indoor kitty because this cat has more places where the tyrosinase enzyme is not active because it's warmer. Here, your outdoor kitty, it gets to go in the chilly cold of the morning and the evening. And so this part of your kitty is colder more often. And so it has more pigment because it's cold enough for the tyrosinase enzyme to work. So this is a time I'm going to ask you to think about another question. And I want you to explain why these two kitties look different using your enzyme vocab. Then I want you to predict the color of newborn Siamese kitties. All right. So if you describe it with something like this, the tyrosinase enzyme starts a biochemical pathway allowing fur color to be expressed in cats. It is a mutant enzyme that only functions in colder weather. And so outdoor cats that have exposure to colder weather have more pigment than indoor cats. You're correct. As for the second part, if you predicted the color of a newborn kitty would be all white, you are correct. Look at those little kitties. So the reason these kitties are all white is because they have been in a really warm environment. They've been in the mom's uterus, which is about 101 degrees. That's how hot cat temperature is. And so these little kitties don't have any parts on their body that have been exposed to enough cold yet for the tyrosinase enzyme to work. As this little kitten's nosy here exists in the world and starts to be exposed to chillier temperatures, the tyrosinase enzyme will start to function and you'll get pigment in the nose, in the ears, in the arms, in the tail. All right, let's practice a word problem. Describe in complete sentences how amylase functions using these terms. Amylase is the enzyme that breaks starch into glucose in our digestive tract. The substrate is the molecule that the enzyme works upon. So the substrate is starch. The product is the molecule that the enzyme ends with that's being produced. That would be glucose. The active site is the location on the amylase enzyme where the substrate binds. The substrate binds to the enzyme with a very particular fit. It's kind of like a lock and a key. It's super specific. And as the substrate comes closer to the enzyme will bend it a little bit, kind of pulling on it, making sure that it fits right in, kind of like clicks into place. That process is called induced fit. The reason that our enzymes use induced fit is because they only want to work on certain substrates. Our enzymes are really picky about what they will work on. Once the substrate is bound to the enzyme at the active site, the enzyme will lower the activation energy needed to turn the substrate into the product, which means that enzymes allow the reaction to happen quicker and cheaper than it would without the enzyme. So activation energy is the amount of energy you have to put into a chemical reaction for it to occur. And enzymes lower the activation energy of chemical reactions. Let's do another one. Describe in complete sentences how amylase functions using these terms. Kinetic energy is the energy of movement. Potential energy is stored energy. And chemical energy is potential energy that's being stored in chemical bonds. We eat food because we're going to use the chemical energy in it. So the chemical energy in this case is in the bonds between the monomers of my starch. When I break apart my starch into glucose, I am using a catabolic reaction because a catabolic reaction breaks things into pieces. If I were building starch, it would be an anabolic reaction. I remember this because I think anabolic steroids are used to build muscle. And then I remember catabolic breaks things down because I picture a cat sitting on a table knocking a glass off the table because cats break things. So catabolic, you're breaking it. Free energy is the energy that's available to do work. Activation energy is the energy you need for a chemical reaction to occur. Endogonic reaction is a chemical reaction that requires energy. And an exergonic reaction is a chemical reaction that releases energy. Both endergonic and exergonic reactions still require activation energy. I like to draw a graph when I do this in class. I think it helps me understand what's going on. Here we have energy. If you're using it, you go to the top. If you're releasing it, you go to the bottom. You're starting off neutral. Here I'm going to take two things and build them together. This first bump on the blue is the amount of energy you need to start the reaction with an enzyme, our activation energy. Here on the red, this is the amount of energy you would need to start this reaction without an enzyme. So using an enzyme lowers the activation energy. I'm going to take A and B, I'm going to bind them together, and when I do, I'm going to end up down here, which means I released energy. So since energy is being released, you left off with energy being put out. That's an exergonic reaction. I took two things and I bound them together, so that is an anabolic reaction. And here I've got energy in the bonds, so it's chemical energy. Let's practice a multiple choice question now. So lactase is the enzyme that breaks lactose sugar apart into smaller sugars during digestion. What is the substrate in this reaction? Lactase ends in ACE, so that's the enzyme, and it tells you ACEs are enzymes. It breaks lactose sugar apart, so it's working on lactose sugar into smaller sugars. So the substrate is the lactose sugars. If I asked you what the product was, you would say smaller sugars. And if I asked you what the enzyme was, you would say lactase. Because lactase enzyme can break down lactose sugar, it is likely also to be able to break down many other types of sugar. This is false. Every enzyme only works on a specific substrate. So lactase enzyme can only break down lactose. Pepsin can only break down protein. Amylase can only break down starch. It's very specific. Every time your body needs to break down a molecule of lactose, it must produce a new lactase enzyme. This is also false. Lactase enzymes are not used up in the process. They are totally recycled. They don't die. You have to inactivate them if you want them to stop working. So your lactase enzyme can continue functioning every time that you eat lactose, if you eat lactose. Denaturing an enzyme means that it has Denaturing means that it has lost its 3D structure, which means that it's probably not functioning very well if it's functioning at all. And our last question, the optimum pH of the green enzyme is closest to, for all y'all that are colorblind, this one here, the first one is green. The optimum pH of an enzyme would be where the rate of reaction is highest, where it's doing the most of its work. That would be the peak of this point. It looks to me like the peak is just about over 2, maybe like 2.2. The optimum pH for the red enzyme would be over 7. So the correct answer for this one, for the green one, is B2. All right. Thank you for watching the lecture. We're going to have questions like these on the exams that are coming up. So study them. Let me know if you feel comfortable with them. If these felt like really hard questions, might be a good opportunity to do the homework again, read the textbook again, practice a new way of taking notes. And as always, email me because I love emails. So hopefully I will talk to you soon. Have a good day.